Hello and welcome to another video in our course on cell biology and genetics. In this video, we're going to talk about the genetic code. So what are you going to learn in this video? First, I'm going to explain what the genetic code is and how it works. We are going to review what codons are, and I am going to talk about the most important characteristics of the genetic code and what it means that it's non-overlapping and degenerate. After watching this video, you will also understand the terms stop codons and sense codons. By the way, you can find written notes on what we are going to discuss today at medicinecanbeeasy.com. The link is also in the description. Okay? So what is the genetic code? Genetic code is the language, set of rules or instructions that the organism uses to translate information in a gene into a protein. It is the means by which it is possible to synthesize a protein based on the information stored in a gene. The code uses codons, sequences of 3 nucleotides, which specify the amino acid sequences of proteins. To understand how the genetic code works, let's review the process of synthesizing a protein. To synthesize a protein, the nucleotide sequence of the gene that codes for a protein is transcribed into mRNA. This is called transcription. Then, the information is translated on the ribosomes into the actual amino acid sequence of a protein, with each group of 3 nucleotides, a codon, specifying 1 amino acid. This is called translation. This set of rules by which we transcribe and translate the information in a gene is the genetic code. Now, each nucleotide can have one of 4 possible bases: adenine, guanine, cytosine, and uracil, or thymine in DNA. For that reason, there are 4 times 4 times 4 codons. That means 64 possible codons. 3 of these are stop codons that give a signal to the ribosome to stop translation. The other 61 codons are called sense codons because they specify amino acids. And here you can see the table of the genetic code with all the codons and the amino acid they specify. Good. So now that we know what the genetic code is, let's look at some of its important characteristics. The first one is that the genetic code is composed of codons. As we said, a codon, a set of 3 nucleotides in mRNA, specifies 1 amino acid that is going to be present in the protein. For example, the codon of guanine, cytosine, uracil codes for alanine. The second important characteristic is that the genetic code is non-overlapping, which means that each nucleotide is part of a single codon. In other words, one nucleotide belongs to just one codon. For example, a sequence of cytosine, uracil, guanine, guanine, adenine, cytosine is going to code for 2 amino acids, leucine and aspartic acid. However, if it were overlapping, it could be read as follows: leucine, tryptophan, glycine, and aspartic acid. So we have to remember it's non-overlapping. The third one is that the genetic code is degenerate. This means that amino acids can be specified by more than just one codon. For example, alanine can be specified by the following codons: guanine, cytosine, uracil, or guanine, cytosine, cytosine, or guanine, cytosine, adenine, or guanine, cytosine, guanine. And this makes sense considering that we have 64 possible codons and only 20 different amino acids commonly found in proteins. The codons that specify the same amino acid are called synonymous codons. And finally, the genetic code is almost universal, which simply means that each codon specifies the same amino acid in almost all organisms. Some exceptions, however, apply, for example, in mitochondria. Great, so this is it for the genetic code. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos on genetics or medicine in general, please subscribe. Thank you again and see you soon.